Welcome back. We are discussing the lesson Human Health and Diseases. In the previous session, we have discussed about some of the very commonly occurring diseases in human beings caused by bacteria, virus, fungi, protozoa and worms. Continuing the series of discussions, let us talk a little about two of the deadliest diseases found in human beings, that is AIDS and cancer. Let us first start the discussion with AIDS. AIDS is an acronym for Acquired Immunodeficiency Syndrome. So, as the word itself suggests, this is a disease that is acquired by a person during his or her lifetime, meaning it is not present by birth, it is not congenital, but it is acquired. And what does this particular condition do to a person? It makes their immune system weak. So that is why acquired immunodeficiency, the immune system becomes deficient, so it becomes weak. Therefore, the immune system is not able to protect our body from some of the pathogens. And the last word is very important here, that is syndrome. So, AIDS, it is not a disease nor is it a disorder, but it is a syndrome. A syndrome is nothing but a collection of symptoms. So many symptoms, when they occur together, that will result in the condition called as AIDS. So AIDS, the causative organism, is a virus. In human beings, it is caused by a virus called as HIV virus, that is human immunodeficiency virus. It belongs to a group of viruses called as retroviruses. These retroviruses, they contain RNA as their genomic material. So we have DNA as our genetic material. These retroviruses which cause HIV/AIDS, they have RNA as their genetic material. So let us discuss about how does a person acquire this disease? How is this disease transmitted from a infected person to a healthy person? Are there any techniques or technologies to detect AIDS in a person, and what are the preventive measures and cure available for this disease? So since it is a viral disease, it is an infectious disease, it spreads from one person to another. So the means of transmission or means of spread, so mostly it is spread by sexual contact, that is unsafe sexual contact. So people who have multiple sexual partners, they are at high risk of acquiring this particular syndrome. So unsafe sexual contact, that is without the use of any contraceptive devices, the virus can spread from the infected person to a healthy person. Through blood transfusion, people who are in need of blood transfusion regularly, so such people are at risk. Nowadays this is not so, because the blood banks, they have very strict regulations while accepting blood from donors. So we have to fill up a form and mention whether we have any ailments or do we have HIV AIDS or not. So voluntarily we have to tell it in the form. So such persons, they will not be able to give blood. So blood transfusion is one. Another way is through, it is seen in drug addicts, because drug addicts, as you know, they use a common syringe or needle for taking the drug intravenously. There will be a group of people who will be sharing drugs, and all of them will be using the same syringe, most probably than not. If one of the person is infected, then the infection can pass to all the people who are involved in this process of taking drugs. So drug addicts are also at high risk. This is also spread from mother to the child. So if the mother is HIV positive, means she has the virus, it can spread to the child through the placenta during pregnancy. So these are the most common methods by which the infected person can transmit the virus to a healthy person, and the high risk candidates are also mentioned over here. So once the virus enters into the body, what happens? What does the virus do? So there is a beautiful flow chart given in the NCERT textbook which explains how the virus goes forward. So first what happens is, once the virus enters into the cell, it attacks the macrophages. Macrophages are one of the soldiers of the immune system of the body. So these are cells which are involved in destroying the bacteria and other pathogens. So the HIV virus, which will infect the macrophage. So when you see the flow chart, we can see that the virus, it will approach the macrophage. Then once it attaches to the macrophage, it will insert its genetic material, that is its RNA, into the cell, into the macrophage. From there, the RNA is then converted into a viral DNA using the enzyme reverse transcriptase that is present in the host cell macrophage. So the viral RNA is now converted into viral DNA. This viral DNA, it will then incorporate itself with the host DNA. So inside the nucleus we have the host DNA. The viral DNA and the host DNA, they become integrated or incorporated with each other, and from there the viral DNA, it will control the production of new viral particles. So production of new viral particles is controlled from the host DNA. So in this case, the host is not able to recognize and differentiate between the self cell and between the foreign pathogen, that is the virus, because of which the new virus particles are created. When the number of virus particles will increase, they will rupture, they will burst open the macrophage and viruses are released into blood. From the blood, these viruses will attack the helper T cells. T helper cells play a very important role in the maturity and activation of B lymphocytes and the T lymphocytes, which play a very important role in immunity of the body, the T helper cells. So the newly released virus particles, they attack the T helper cells, and from the T helper cells they once again produce new virus particles. In the T helper cells, and once again when the viral load increases inside the T helper cells, the T helper cells are once again burst open, they are destroyed. So what happens is, as the condition progresses, as the virus keeps on reproducing, multiplying in number, the T helper cell numbers will start decreasing. The number of T helper cells will progressively, it will come down, because of which what happens is the immune system, it becomes weak, because these are very important in maintaining a good immune system, maintaining the immunity of the body. So because the T helper cell count decreases, what happens is the body is susceptible to even minor infections. So infections caused by bacteria, other virus, fungi, and also certain toxoplasma, they can become bigger because the immune system is weak. It is not able to protect the body from these infections. So what happens is a minor cold may turn into pneumonia. So it can affect the respiratory system. A minor fever can turn into a deadly fever. And there is always a time lag from the time when the virus enters into the body and the time when the symptoms show up. So there could be a time lag of anywhere between 5 to 10 years from the time of infection till the time the symptoms show up. So as you know, AIDS is a collection of symptoms, it's a syndrome. The common symptoms of a person infected with HIV is fever, then weight loss. This is a very observable feature, and diarrhea. So these are the common symptoms, and as you can see, you cannot differentiate this from a normal person who may not have AIDS. As the number of virus particles increase in the body is more susceptible to infectious pathogens. So the immune system will weaken and new infections will occur in the body. So what is the mode of diagnosing or detection? How can we detect the presence of virus? There is a universally accepted diagnostic technique called as ELISA, that is enzyme linked immunosorbent assay. Assay means a test. So this test will tell us whether the virus particles, that is the HIV virus, is present inside the body or not, and it will also give you the quantity of virus particles present. It is a very accurate assay and the results will come out in a very short period of time, that is in a few hours. And as I already told you, the time of infection and the time symptoms start appearing could be delayed as long as 5 to 10 years also. So what is the cure available? Nowadays antiviral drugs or antiretroviral drugs are present, this will be able to reduce the number of virus particles up to a certain amount, but it cannot completely get rid of the virus particles. So what happens is, a person who is consuming these retroviral drugs, they can increase their life expectancy. The longevity of the person will increase, but the disease, the syndrome cannot be completely cured. So prevention is the best way in order to not acquire this virus. And a lot of initiatives are taken by World Health Organization, the other non-governmental organizations, and in India also we have an organization called as NACO, which is responsible for keeping the syndrome under control. The AIDS was first detected in the year 1981, and from then it has known to have taken more than 25 million lives. That is a huge number. Therefore, it is a very serious disease which needs to be controlled, and measures should be taken to prevent. Prevention measures include, number one, creating awareness among people. So therefore, it is involved in textbooks, in schools and in colleges, and that is precisely why we are learning about this in the second year PUC class. So creating awareness among students and the general public about the mode of transmission and how it is spread from one person to another person. Most probably it is spread by the exchange of body fluids. So it is not spread by physical contact, just by touching a person or sharing their utensils. So usually what happens is, there is a stigma in society which will try to isolate people who are HIV positive. So such people should not be isolated, and with proper physical fitness, exercise, medicine, diet and psychological counseling, they can lead a normal life. So creating awareness is the first step. Second, there should be regular health checkups in high risk populations in both government hospitals and clinic. So populations which are suspected to have more number of HIV positive people, in such populations a regular health checkup can detect the disease at an early stage, and proper curative measures can be put in place. That is one. And also using disposable syringes and needles, this is followed everywhere nowadays. So in the olden days, the same needle was boiled and then it was used for other patients, but nowadays we have disposable syringes and needles. Once used, they can be thrown out. And also free distribution of contraceptive devices, so that people free distribution of condoms. So there is a way to prevent acquiring this particular disease. So these are some of the common techniques. And also in blood banks, so blood banks have measures in place which will check the donor before they donate the blood. So prevention is better than cure, and we know that for AIDS there is no cure available. Therefore prevention is the only way to prevent acquiring this disease. That is all about AIDS. how it is caused. It is caused by a retrovirus called as human immunodeficiency virus. It is spread by sexual contact through unsafe blood transfusion, commonly seen in drug addicts who use a common needle, and from the mother to the child. It affects the macrophages, the T helper cells, therefore the T cell count will decrease in the body, so the body is susceptible to minor infections which can become major. Symptoms are fever, weight loss and diarrhea, and it is detected by ELISA test. Next, let us discuss another disease which is a non-infectious disease responsible for the death of millions of people, which is cancer.